Okay, welcome to Chapter 7. I hope everybody is doing fantastic today. In this chapter, we are going to be talking about cell signaling, basically how cells communicate with each other. And so we'll go over some of the basics. This may be a new topic to you. Talk about some examples of how signals are perceived and propagated by cells. Um, and then at the end, we'll talk a little bit about what the end result of these signals might be. So how would these signals result in changes within cells? And I'm going to have this split into two parts. And this whole idea of cell-to-cell signaling or communication, of course, it can be important for single-celled organisms to be able to communicate with each other in some way. But this whole process is really, I think, arguably most important in multicellular organisms. If you think about your own body, you have cells within a single tissue that may need to be coordinated and be able to communicate with each other, or even cells in totally different parts of your body that have to be able to signal each other. And as I'll talk about, um, a lot of what we've learned about this process of signaling comes from trying to study human disease. So, for example, cancer is often a disease that's caused by changes in these cell signaling pathways, specifically in signals that would help drive the process of cell division, mitosis, which we'll be talking about in an upcoming chapter. So if these signaling pathways are defective or basically hyperactive, you end up with too much cell division and hence a tumor. So trying to understand the cause of cancer, the cause of tumor growth has kind of led us into an understanding of these signals and these pathways. So if we think about this kind of big picture, all cells have to be able to process and receive information from their environment, and that information could take on different forms. It could be some physical stimulus like pressure, could be a light signal, but in this chapter we're really going to focus on chemical signals, so cells perceiving some other molecule in their environment. Um, and typically, as I've already implied, these signals are going to come from outside the cell or potentially even outside the organism themselves. So it's some external signal. And in order to be able to perceive any signal, whether you're talking physical, chemical, light, whatever the case might be, there has to be some mechanism to perceive it, right? We, as humans, we can't receive light signals without our eyes, right? We need that sensory organ. We can't receive sound signals without our ears. So it's the same idea even at the cellular level. If a cell is going to try to perceive a chemical signal, there has to be some mechanism to do that. And you'll see this term throughout the chapter talk about signal transduction pathways. And basically, I want you to think of these signaling events almost as relay races, I think is a good analogy, where you have one runner in a relay race, right, passing the baton to the next, who then passes the baton to the next. So you can think of these as pathways because there's going to be multiple molecules involved, and they are going to be passing that signal, passing that baton from one to another. Sometimes you may also see this referred to as a signaling cascade. There's three main sort of levels, I guess would be the best word to use, levels of signaling that I want you to be aware of: autocrine, paracrine, or hormone signaling. And the difference between these really all comes down to how the distance that the signal is traveling. So auto always means self, and an autocrine signal is an instance where a cell is actually producing a chemical signal. So this cell you see here in this image is producing this, let's say it's a protein, that's the signal, right? It's exocytosing it, and then that signal is now being perceived by the same cell that produced it. So it's almost like you're calling yourself on your cell phone or texting yourself in some way. You're sending yourself this message. Um, and so there are all kinds of examples of this happening in your body. Autocrine, almost self-signaling. And then we have paracrine signaling, which is just short distance local signaling, where one cell produces the signal, and then that signal diffuses away from that cell and is perceived by other cells in the vicinity. And you can see in this example we have the cell at the top here that actually has a receptor. This goes back to the eyes or the ears, the capability of perceiving a signal. So this cell has the receptor to actually receive that signaling molecule. So it gets the message. This cell down here, even though it is in the neighborhood and it is kind of being bombarded by these signaling molecules, because it doesn't have the receptor, it's not going to receive that signal. But this would be an example of paracrine signaling up top. So this cell on the right is being signaled by this cell on the left, short distance signaling. And then lastly, we have hormone signaling, which is used in multicellular organisms to transmit long-distance signals. And hormones, as we've talked about, are usually small lipid molecules that can diffuse across membranes pretty easily in most instances. And hormone signals may be produced in one part of the body and actually transmitted through the circulatory system to, all parts or distant parts of the body. So long distance signaling in a nutshell. And I mentioned that these signals, these pathways, are sort of like relay races. So here's what I'm talking about. So here's kind of a basic overview of sort of a typical signal transduction pathway. So this is always going to start with some chemical signaling molecule in this case, and then that has to be perceived by some receptor protein usually on the surface of the cell. Right, so we have the baton being passed here. The binding of the signal to the receptor activates the receptor. That's what this little yellow flash is supposed to indicate. Causes a little shape change in the receptor, so it becomes activated, biologically activated. And now that receptor is going to pass the baton, pass the signal on to another molecule in this pathway that's now inside the cell. So this protein interacts with the receptor in some way, and it becomes activated. And then it may pass the baton on to more downstream runners, and so on, until eventually we are going to cause some physiological change in that cell. And there's different things that may happen, but one common event that's being depicted here is this signaling pathway is going to lead to the nucleus, and we're going to basically alter the transcription of certain genes, and that would allow us to either stop producing proteins that we're already making in the cell, or maybe produce new proteins that weren't previously being made in the cell. And the presence or absence of those proteins then are going to change the biology of that cell. It's basically the outcome. Okay, so let's go into a little bit more detail here, but hopefully you have this basic idea of, signal binds to receptor, receptor activates one molecule, which activates another, and then eventually we get some outcome, some longer term change occurring in that cell. So I think this slide is just kind of summarizing what I was just talking about on the previous slide. So I'm going to go over that, or skip over that, but you've got that for your notes. So let's focus in on the first step in this pathway, which is ligand signaling molecule binding to receptor. So ligand is just another term for that chemical signaling molecule. And because the ligand, that signal, is usually coming from outside the cell or maybe even outside the organism, um, the receptor to perceive that chemical signal has to be on the cell surface in most cases. So you can see here the brown structure here that's embedded in the membrane is a receptor protein. And this would be an example of an integral transmembrane protein. You can see the receptor goes all the way through the membrane. Part of the receptor is exposed on the outside of the cell. That's the part that would actually interact with, bind to the ligand. And then part of that receptor is inside the cell. Okay? And so we'll get this physical binding, but that binding is going to be usually pretty weak and temporary. Um, so the ligand could bind for a short period of time, but then it's going to be released, fall off. And so the temporary nature of that interaction is really just to ensure that this signal is not constant, right? Usually we want that signal to be perceived, but then we don't want the response to go on forever, right? And so in terms of biology, these receptor ligand interactions are really critically important. So these interactions are happening all the time, and if any of you are pre-pharmacy students, um, I wanted to point out that a lot of drugs work by interfering or maybe even enhancing in some way these receptor ligand interactions. So just to give you one simple example, um, most of us probably take in some caffeine most days, and caffeine works by, it is one of these ligands. It binds to a receptor on your cells, um, and basically blocks the receptor from being able to bind to its normal ligand, which would be adenosine. So in your normal situation, um, basically adenosine binds to these receptors on nerve cells, and it then initiates this signal transduction pathway that is going to the end result is going to be to reduce brain activity, and it's basically your brain's way of telling you, hey, you're tired, right? It's the end of the day. You need to start shutting things down and go to sleep. Well, if you take in some caffeine, caffeine will now go, and because of its similar molecular shape to at least part of it, the adenosine molecule, it can bind to that same receptor that adenosine would bind to, but it doesn't actually activate the receptor, it just binds and kind of blocks adenosine from being able to get in. So you're going to feel less tired. But that's just one example. There's all kinds of um, known drugs that either bind to and activate or block different receptors. So I mentioned these receptors are almost always going to be at the cell surface. Really the one exception, so there are some cytoplasmic receptors inside the cell. Really the one exception is for receptors that are intended to bind hormone signals. So we've talked about the fact that hormones are usually small nonpolar molecules that can actually diffuse right across the membrane. So for those hormone signals, they don't necessarily have to bind to a receptor that's outside the cell, right? They can actually go into the cell and bind to a cytoplasmic receptor. Now, I want to show you three different common types of plasma membrane cell surface receptors. So these are sort of big families, and they're very well understood and studied. So I just wanted to show you kind of a generic overview of these three different types. And they are what are called ion channel receptors, protein kinase receptors, or G protein linked receptors. So let's start with the ion channel receptors. And as that name implies, these receptors can actually act as membrane channels that would allow ions to cross the membrane. So you may remember in the last chapter we talked about channel proteins and the fact that you could have gated channels. And that's basically exactly what these ion channel receptors are. They are gated channels. And so by default, the gate is closed if the appropriate signaling molecule or ligand shows up. In this case, it's this neurotransmitter called acetylcholine. It can bind to the receptor. That causes a shape change that now opens up the channel. And in this case, this channel is really meant to allow sodium ions to flow across the membrane. And this particular example, this is what would initiate a muscle contraction. So you would imagine this is a muscle cell, and you have a nerve, maybe coming from the top of the image here, where the nerve ending is sitting right near this cell. So your brain sends a signal down that nerve that is intended to cause this muscle to begin contraction. That nerve ending will release the neurotransmitter acetylcholine, which will diffuse across the synapse and bind to these receptors on the muscle cell. So this would be an example of like paracrine, short distance signaling. Acetylcholine binds, the channel opens, these sodium ions that are more concentrated outside the cell now flow into the muscle cell, and now that's going to trigger changes in the muscle cell that are going to lead to that contraction. So this big class, there's lots of different examples of ion channel receptors. The second common type of receptor, what are called the protein kinase receptors. And protein kinases are enzymes that are capable of phosphorylating other molecules. So in this case, the receptor is actually a protein kinase enzyme. So once activated, it can then begin to add phosphate groups to other molecules inside the cell. And what you see in this example is the protein kinase receptors, when they're inactive, they usually exist in two pieces, and the ligand, the signaling molecule, would bind to both pieces and bring them together, thereby activating that receptor. And we can see in this case the signaling molecule is a growth factor. And so once that receptor is activated, now it's going to start collecting phosphate groups. Right, so since it's a kinase and it wants to add phosphate groups to other molecules, it needs some phosphate groups to be able to do that. So once it's collected enough phosphate groups here on the portion of the receptor that's inside the cell, now it becomes fully activated, and it will be able to start passing some of those phosphate groups off to other molecules. Okay, so in this case, you can think of the phosphate groups in this particular pathway almost as that baton that's going to be passed from one runner to the next in the relay race. So our protein kinase receptor then activates this protein called RAS, and you don't have to memorize all these names. RAS activates another protein kinase called RAF, which can then phosphorylate another protein kinase called MEK, which can then phosphorylate another protein kinase called MAPK, and so on. So you get this passing of that phosphate baton through this phenomenon that's called a protein kinase cascade. So you basically have one protein kinase activating another. And then eventually this pathway will lead to the nucleus, and we'll get our cellular response. And the cellular response in this case, because our signal was a growth factor, is going to be growth, aka cell division, right? So this whole pathway would be designed to initiate the process of cell division and mitosis. Now, just to focus in on that, let me go back here, on this idea of the kinase cascade, you might be looking at this and think, well, this just seems crazy and inefficient. Like, why are there so many steps here? If the goal is to get this cellular response, why don't we just have, okay, growth factor binds to receptor, and then maybe we activate like one protein here in the cell, and that can just go in the nucleus and give us that response. Why do we need this whole cascade of kinase to kinase to kinase? And the short answer is there's really two reasons for that. So all these different steps in that cascade amplify the signal, make it larger. Okay, so you could imagine, let me go back here, you could imagine, one of these RAF kinase molecules may be able to activate, 10 MEKs. So now you went from one RAF that was activated to 10 MEKs, and then each one of those can activate 10 MAPKs. So if they all do their job, you're up to like 100 of these active molecules. So it's basically just making the signal bigger and bigger and bigger with each pass of the baton. And that's useful because it means the cell is not going to be able to miss or ignore that signal, right? We just had one lonely growth factor show up at the surface of that cell, but by the time we've run through this kinase cascade, we've got this, army of activated molecules marching into the nucleus, and hey, you better start mitosis right now. Get things rolling. So we make the signal bigger so the cell doesn't miss it. And then all these different steps also just provide a lot of different control points, so a lot of specificity in terms of how a cell might be able to tweak that pathway or even branch it off into another signaling pathway. So amplification and specificity are really the big advantages of these, all these relay runners, all these different steps in these pathways. And as I mentioned in the introduction, in particular with this type of protein kinase receptor pathway that leads to cell division, right? If something becomes overactive in this pathway, you end up with too much cell division and a tumor and potentially even cancer. So as I said before, right, a lot of what we understand about these pathways is because of our desire to better understand cancer. So we had our ion channel receptors, our protein kinase receptors, and the third and final kind I wanted to mention are the G protein linked receptors. And these are so named because the receptor is linked to or interacting with a protein called the G protein. So you can see here's the receptor, right? Cell surface receptor. The G protein is just a peripheral membrane protein, so it's just bound to the inside surface of the membrane. And you can see the G protein actually has a few different pieces or subunits. And the default state is that G protein is inactive. And it's inactive when it is bound to this molecule called GDP, guanosine diphosphate. This is similar to ADP, adenosine diphosphate. That would be a precursor of ATP. So it's sort of an energy molecule precursor. But bottom line, once this signaling molecule binds to the receptor, now we get a shape change in the receptor that is going to cause the GDP on the G protein to now be swapped out for GTP, which, as I just said, is an energy-rich molecule very similar to ATP. Now that G protein is activated and it can diffuse off down the membrane, move down that inner surface of the membrane, or at least a subunit of it will move down the membrane, taking the GDP with it until it bumps into the next runner in the relay race and activates it. And then we're into that whole cascade or relay race at that point. And once this downstream protein is activated, you'll notice GTP is converted back into GDP, and the signaling molecule, the ligand, falls off the receptor, and we're basically back to our starting point. And here's what that whole process would look like for a cytoplasmic receptor. As I mentioned, there are cases where hormones, in this example cortisol, can diffuse right across a plasma membrane into a cell and bind to a receptor inside the cytoplasm. Um, so in this case, the cortisol receptor is held in this inactive state by this binding partner called a chaperone protein. But as soon as cortisol shows up and binds to the receptor kicks the chaperone to the curb there. That receptor becomes activated, and in this case, the receptor actually can directly move into the nucleus, then bind to the DNA and alter transcription. So in a sense, this would be a much simpler process, less steps involved anyway. Okay, so all these examples we've talked about so far, the ligand, the signaling molecule would be considered the first messenger in this pathway, right? It is bringing the signal to the cell, binds to the receptor, and triggers this whole relay race, this whole process. But what I want to talk about in the second half of chapter 7 is what are called second messenger molecules. And by definition, these second messengers are always small non-protein molecules um, that are found inside the cell. And basically, the advantage to these second messengers is because they're small, they can diffuse into all the corners of the cell and basically make sure that that signal is getting everywhere it needs to go. And in that process, they're also helping to amplify the signal. So in the second half, I will show you some examples of some second messengers and how they fit into this whole process of these signaling pathways.